welcome everybody. This is the, what are we calling it, guys? The Outer Circle Catchment Area of Ontario. I think Jagdeep had actually come up with a better name than that, for Genome-Wide Sequencing Ontario. And it's, hopefully we have some folks on from our various sites, and because today what we want to do is sort of talk a little bit more about the logistics. And so I'm just going to open and close this session, but Meredith and Caitlin will actually be doing the bulk of the workflows in terms of how we're going to start to move samples forward. So just to recollect, I think maybe, what was it, 2 weeks ago, we had a more generalized information session, and we talked about, you know, how we arrived here in terms of our current landscape of genome-wide sequencing, and how this project we're going to describe today is actually got the words project associated with it, or pilot, in that it is really a two-year defined piece of work that we're working on closely with the Ministry of Health and producing outcomes for them so they can make informed decisions about how this will continue in Ontario. So we did some introductions, but today what we're going to do is focus on how we are going to order whole exome sequencing and all the details related to that, some of our data analysis and results interpretation, what we're going to report, and then I'll finish up with just talking a little bit about some of our research opportunities and why they're important and why we need, you know, excellent engagement from all of our centers. And then there'll be some spots that if you don't have a chance to get your question in today or have clarification, how you can do that. So just to remind everyone, as I said, this is a pilot, and it is a pilot for 2 years in partnership with SickKids and the Ontario Ministry of Health. We also have additional support from Genome Canada and Ontario Genomics in the form of a Genomics Application Partnership Project grant, which we actually received several years ago, and it took this long for us to get to this April the 1st launch. So we're super excited about that. Just to recollect, though, this funding from Genome Canada has not been formally announced and is still under embargo, just for this audience. So if there's questions as we go through, just type them into the chat, and there'll be a little bit of time hopefully at the end, and also some ways that you can contact us if you have questions we don't have time to get to today. Okay, so over to you, Mare, I think.
Yeah, so thanks, Kim. So over the next couple of slides, I'm just going to be going over the process of ordering genome-wide sequencing through GSO. So this will include some discussion around the eligibility and the process of having these cases reviewed, all the different required documents, the consenting process and the options that are available to the patients. And then Caitlin will start talking about the sample types we accept and going into the analysis piece. So just a reminder to those that attended the last session, and new information for those that are just joining us for the first time today. GSO is going to be focused on clinical genome-wide sequencing that is being requested on a routine turnaround time. So any prenatal and expedited postnatal requests will continue to be reviewed by the Labs and Genetics branch at the Ministry of Health. They'll still be doing the prior approval process and will be sent out of country. Over the course of this 2-year pilot, genome-wide sequencing is going to be offered in the form of both exomes and whole genomes. So the majority of patients are going to be receiving whole exomes over the course of the project, with a subset of 650 trios from CHEO and SickKids that will be randomized between either exome or genome. So this stratification will be important really in informing the ministry about the diagnostic and clinical utility of genomes compared to exomes, and help them in making determinations around if and when genomes should be offered on a clinical basis more broadly to people in the province. Both CHEO and SickKids GDLs are involved in this project and are going to be offering genome-wide sequencing through a hub and spoke model, which we'll be discussing further in a few minutes. In order to properly build capacity, and Ontario genetics clinics were split into catchment areas. So either sending to SickKids or sending to CHEO. And these catchment areas are defined by the clinic addresses, not the patient address. So in preparation for the launch of GSO, we completed a review of provincial test volumes for all of the clinics over the past several years to get an idea of how many exomes were being ordered by each clinic. And based on that, we split the areas into a 2/3 and 1/3 split. So SickKids is taking on 2/3 of the provincial volume, so focused on the GTA. And then CHEO is taking on 1/3 of the provincial volume. And like Kim said, this kind of works out to the outer perimeter of Ontario. And while there are 2 laboratories reporting out the results of genome-wide sequencing, all patients are going to follow a unified workflow that has been built by both CHEO and SickKids teams at every step. So one difference between GSO and the out-of-country program will be that providers are not going to be applying for pre-approval to the MOH prior to ordering. Rather, samples will be sent along with their requisitions directly to the GSO laboratory of their catchment. So for everyone on this call, you'll be sending everything directly to CHEO. DNA is then extracted at the CHEO lab, and we will be sending an aliquot of the DNA to SickKids, who will then take over the role of doing the sequencing and running it through the bioinformatic pipeline. Data is then going to come back to the CHEO site and be analyzed by CHEO, and all reports will be issued from CHEO as well. If there is ever any segregation testing that needs to be done to clarify the classification of a variant, that's also going to be done locally at CHEO. So over the next several slides, we're going to discuss the different components necessary for a request for genome-wide sequencing to be processed and approved by the GSO laboratories. So this is going to include discussions of the requisition, Ministry of Health criteria for genome-wide sequencing eligibility, clinical documentation, consent options, and the necessary samples. All forms we discussed today are currently in the process of final institutional approvals, and they're going to be available to everyone via the GS Ontario website prior to April 1st. So that's gsontario.ca. So when ordering genome-wide sequencing, a requisition is going to be required for every individual that's submitted to CHEO. So GSO has developed 2 different requisitions to facilitate this. So the first is the more in-depth requisition, which is for the proband. And that's going to include standard demographic information, look very similar to other requisitions you've seen before, and will include the information for the referring geneticist and genetic counselor. There will also be a clinical data sheet for documentation of patient phenotype and family history, and the clinical genome-wide sequencing testing criteria and attestation form, which is going to look very similar as it is built off the same criteria that the Ministry of Health is currently using to facilitate out-of-country exome. And then finally, there will be a consent and acknowledgement form for families to fill out to document their consent choices, as well as an information sheet attached to every requisition that the family can take home with them. The family member rec is much shorter. It will just contain only the demographic page as well as a clinical data sheet, so that if there are any additional phenotypes or there's another additional person in the family that's affected, that can be documented there. And as has been current practice, clinical notes, letters, and relevant reports on the patient must also be provided along with the sample and requisition. Here we've included just a few shots of what the requisition looks like to start familiarizing yourself, but no need to focus on it too much, as we'll make sure that the finalized forms are available to you well in advance of the launch of GSO. So here we just see that first page, the demographic and healthcare provider page, as well as that clinical criteria attestation form. When you are starting to review the forms, there are 2 differences that you will note on the provider attestation form, and those are located at the top and the bottom. At the top of the form is a patient summary section, which will capture the proband's phenotypic category, their age of onset, ethnicity, and previous test history. And this section really serves two purposes. The first is going to allow for the stratification between either genome or exome for that smaller SickKids and GEO cohort to ensure we have comparable individuals in both groups. But also it's going to help us to look at and assess the efficacy of the MOH criteria for different patient groups. And then at the bottom, you'll see a prompt regarding Care for Rare Solve Phase 1. So GSO has chosen to include this prompt as the clinical utility data captured through Care for Rare is going to be really important to informing the Ministry of Health about making these downstream decisions about whether or not genome-wide sequencing is ready to be incorporated into clinical care. All right, so next we're going to talk about the eligibility criteria and review. So two main components are going to be reviewed when determining patient eligibility for genome-wide sequencing. The first being the patient's clinical criteria, so presentation and management, and the second being the provider attestation regarding pre-test counseling, clinical assessments, and genetics expertise. GSO is only currently set up to serve Ontario patients with OHIP funding. But of course, in Ontario, we see patients from all over the place that have different insurance coverage. So if you are seeing a patient that meets eligibility criteria for exome and it's something that you think is the best test to offer for your patient, we just ask that you reach out to GEO directly and we can talk about options for your patients. All right. So here we have the clinical custom data sheet that's been built for GSO to help capture patient phenotype. So as I mentioned before, you'll be filling this out for all of your patients, but of course supplementing this with clinical letters, records, and reports, as well as family history. And that clinical information you're providing is really going to be invaluable to the analysis team and be used quite heavily by them to make sure that we are getting the most accurate analysis for your patient. So GSO is going to be using this clinical information to perform phenotype-driven prioritizations of variants identified through our pipeline via the analysis platform we're using for this pilot, and that platform is called Emagen. And Emagen uses an AI-based prioritization algorithm. So really, the richer the data, phenotype data that you're able to provide us, the more accurate and stronger analysis that we can do in prioritizing the genes appropriately. And then, of course, all the additional information you're including in your letters that you're sending to us as well, such as onset and progression, are really important to our team of analysts that are looking through all of this, and helps them provide contextual correlation so that they have a more thorough and nuanced analysis strategy when they're looking at your patient. So as we've mentioned previously, Genome-Wide Sequencing Ontario is going to continue to use the whole exome sequencing eligibility criteria that were approved and have been in use in Ontario for the last several years. So this checklist really includes criteria for clinical presentation, as well as the management impact for an individual patient and the family at large. And we will continue to not offer GWS to patients with isolated mild ID or learning disabilities, isolated non-syndromic autism, isolated neurobehavioral disabilities, or if there's a patient with a phenotype that's highly specific to a known genetic condition in which a more optimized test already exists. In addition to continuing to use that criteria, we're still also going to be following the Ministry of Health guidelines on who can order genome-wide sequencing in the province. So providers must practice in the area of genetics, either as a geneticist or genetic consultant, or work in a clinic where a genetic counselor has really been integral to the care of that particular patient. They need to have expertise in performing clinical genetics evaluation, so looking at the family history, doing a genetic focused medical history, and evaluating any previous testing that may have been completed. They need to have expertise in determining whether or not genome-wide sequencing is the appropriate test for this patient, and if it's not, ordering the one that is more indicated. as well as be able to provide adequate pre-test counseling. So talking about the different findings that could come up, including the primary and secondary findings, and have the ability to interpret the results of the clinical genome-wide sequencing and provide adequate post-test counseling to the patients. All right, so the final thing I'm going to be speaking about today before passing the controls over to Caitlin is the GSO clinical consent forms. So our forms were designed after extensive review of the consent forms currently in use at a variety of different labs, as well as a thorough review of the literature to better understand patient preferences for clinical consent. So GSO has two main forms for patient consent. The first is the acknowledgement form and consent form, which is pictured here, which will be used to document patient and family members' decisions around clinical data sharing, secondary findings, and future contact for research participation. The second is that information sheet that I mentioned earlier, and this can be taken home by the family and will outline the purpose of genome-wide sequencing, how it's performed, what is reported and what is not reported, any limitations, potential risks, confidentiality, as well as sample storage and future uses. By signing the acknowledgement and consent form, patients and their families will be indicating that they understand the information outlined on the GSO information sheet and are agreeing to undergo genetic testing to possibly identify the genetic variants responsible for the condition in their family. Patients and families will also be given information regarding the sharing of their clinical data, as well as clinical health information. So anonymized data will be shared with the analysis platform for the purpose of improving the software and in turn providing our patients with a better diagnostic tool. And then anonymized data will also be shared through the means that Ontario Genetics Laboratories have been doing for many, many years. So for the purpose of delivering better care to patients, labs are able to share these anonymized interpretations, for example, through tools like ClinVar. And finally, patients will be informed of the Ontario Clinical Genomic Knowledge Base, which was mentioned in our first talk, which is kind of a first of its kind tool in collaboration in Canada, and is something that we're working very hard to extend across the country. So you'll be hearing more of that in the upcoming months, I'm sure. At this time, the Ontario Clinical Genomic Knowledge Base can only be accessed by the CHEO and SickKids GSO teams. And the granularity of the coded data in that knowledge base is really, we're hoping will greatly improve the filtering, prioritization, and interpretation of variants seen in our patients, and it's going to help improve the test for all. But as this data is coded and shared between two institutions currently, patients and their families will have the option to opt out of this sharing. As we mentioned during the last talk, GSO will continue to offer secondary findings over the course of the pilot. So analysis of secondary findings will only be completed on the proband, but other family members that are submitted for concurrent analysis, so submitted as a trio, submitted as a duo or quad, may have the inheritance of those secondary findings that were identified in the proband reported on them. And Caitlin's going to talk a little bit more about the reporting details in a few minutes. GSO has two secondary finding lists. So the typical secondary finding list that we've been offering for 5 years. So following the ACMG recommendation and offering the list included in the version 2.0 of that list. We are aware of the upcoming version 3 and we'll be assessing it prior to implementing it within the province. And then there's an additional smaller list, which is a list of 52 genes rather than the ACMG 59 that has been assessed by lab experts and geneticists and genetic counselors. And we're determined based on conversations with them and review of the literature to be those genes that are actually medically actionable in childhood. All pediatric programs, so those under the age of 18, will automatically have analysis and reporting of any known or expected pathogenic variants on the medically actionable in childhood list, with the option to opt into the full list. So parents may choose to opt into the full list, or if they are a teenager making decisions for themselves, they could choose that as well. Competent adults also have the option to opt out of the full list, so they can choose to have the full ACMG 59 or choose that they don't want to find any secondary variants. And then family members, as I mentioned before, can choose whether or not to have inheritance reported. And we're in the process of developing information sheets and consenting aids that'll be available on the website to help facilitate this conversation between you and your patients. The decisions around secondary findings were really made based on consultation with providers across the province to better understand the current practice in Ontario. And then we took that information and consulted with legal experts to make these decisions. And Kim's going to speak a bit more to the work that's going to be ongoing over the course of the pilot to better understand secondary findings and their impact on the healthcare system as well as patients. The final piece of the consent form is an option for patients to decide whether or not they would like to be contacted for future research studies. So this could be anything from additional data sharing opportunities, natural history studies, or clinical trials. And patients will have the option to decide how they would like to be contacted and how often they would like to be contacted. And any studies that are contacting patients through this recontact option will need to have RED approval prior to reaching out to patients, and will need to have some sort of plan of how they will involve at least a subset of the patient's healthcare team. Okay. And with that, I'll pass it over to Caitlin.
Thanks, Meredith. Hi, everybody. My name is Caitlin Chisholm. I'm one of the lab genetic counselors at the CHEO Genetics Diagnostic Lab. So if we don't know each other personally, we've likely interacted in that venue at some point in the past. And my part of today's talk is going to go through some of the details of what happens after you have identified an appropriate patient and filled out the forms beautifully, as per Meredith's instructions. And I just want to note, I do see that we have some questions coming through in the chat. We'll have plenty of time at the end for questions, so please keep submitting them, but I'll hold off and address them once we're done the slides. There we go. So in terms of testing strategies, as I'm sure everyone in this audience knows, we really strongly prefer doing a trio test as opposed to a singleton or one parent only. Obviously, that's not always possible, but as you know, the test is most powerful when we're testing both parents. So we would really strongly recommend trying to submit trio samples as much as possible. Of course, when that is not possible, we will accept singleton or duo situations. In some situations, it might be appropriate to test a quad or an atypical combination of family members. And so if you're unsure about the best testing strategy, or if it's a really unique scenario and you just want to talk it through, feel free to reach out to us prior to submitting the samples and requisitions, just to make sure that the people that you're sending samples from are the most appropriate and will give us the best chance of success. In terms of accepted sample types, our preferred sample type is blood in an EDTA or purple top tube. We will accept DNA, provided it's of good quality and quantity. Unfortunately, we are not validated to accept buccal swabs or saliva or other sample types. So really blood and DNA are the ones that are appropriate at this time. If there are any exceptional circumstances, please feel free to reach out to us and we can provide some guidance. In terms of what happens once your requisition and sample arrives, just like all of our other molecular genetic tests, it will be reviewed by a genetic counselor. And the process is the same, whether it's a sample coming from CHEO catchment sites or the Sick Kids catchment site. It gets reviewed. If it meets criteria, we will proceed with testing, and there will be no notification that it is proceeding with testing, just like any other sample. If there's missing information, so if the rec is not complete or we need some more information before appropriately adjudicating the request, you will be notified and we'll be asking you for that missing information. Once the missing information is provided, it'll go through the review process. If the case does not meet criteria, or we're not sure if it meets criteria, it will be reviewed at a joint committee between the CHEO and SickKids team. And this is a committee that includes clinical geneticists, laboratory geneticists, as well as genetic counselors, similar to how the exceptional access review happens right now. After that review, the test could proceed, or it might not meet criteria, and then the test request would be rejected, and you'd be notified if that rejection occurs. If a test request is rejected, people do have the opportunity to make an appeal if there is exceptional circumstances. Just to note that if you know that your scenario is unusual and doesn't meet the strict MOH criteria, but you think genome-wide sequencing is still an appropriate strategy, feel free to provide that additional information at the time of the original request. You don't have to wait to get a rejection letter and then submit it on appeal. So you can submit additional supporting information right from the start. In terms of timing, we aim to have this joint committee meeting once per week. So if you have missing information or the test request is rejected, you should hear back within likely a week to 2, depending on the days and how sample arrival lines up with the review. But it should be a fairly timely notification. And again, if you don't hear anything, that means the test is proceeding as requested. In terms of the analysis and interpretation, just to emphasize that, as previously mentioned, the samples will be submitted to CHEO from CHEO patients as well as from our catchment sites, and an aliquot of DNA will be sent to SickKids. The genome-wide sequencing wet work as well as bioinformatic processing will happen at SickKids, but then the analysis for CHEO samples will happen at CHEO. Both sites are working very closely for this pilot project. So we have common bioinformatic pipelines and annotations. We're using the same software platform with the same set of structures and filters built in. We have the same policies for reporting findings, and we have a shared knowledge base between the 2 institutions. So really, it's the same test coming out of 2 sites. In terms of the actual bioinformatic analytical workflow, this was reviewed at the first information session, but just wanted to highlight, like Meredith said, we're using both phenotype and genotype to do our analysis, so it's important to give us as much phenotypic information as possible. Additionally, prior to the genome-wide sequencing being done, there will be genetic fingerprinting done to confirm family relationships. The analysis will be done in a software program called Emagen, and that's where filtering and interpretation will take place. We will also be storing the data in our local archives. In terms of what's going to be detected, this is similar to what commercial labs are doing currently, so there's nothing groundbreaking on this slide. Just wanted to highlight that these tests will be looking primarily for sequence variants. There are some limitations that we all know for whole exome sequencing, in that the coverage for non-coding variants is not ideal, or not intended to be ideal. While we will be doing a screen for copy number variants, microarray testing remains the standard of care, so this should not be relied upon as a primary test for copy number variants. Additionally, genomic rearrangements, mitochondrial DNA variants, and repeat size variants will not be able to be detected on whole exome sequencing. For the cases that are randomized to receive whole genome sequencing, that has additional power to detect non-coding variants. In the future, we're hoping to optimize to have additional power to detect rearrangements and other types of variants as well. Throughout the 2 years of the project, some of these additional types of variants may come on board. Additionally, our pipelines will be improved throughout the project, so there could be improvements in variant calling and analysis within our term. In terms of reporting, like I mentioned, while the wet work is done at SickKids, CHEO will take care of the result interpretation and reporting for all samples that come through CHEO, and that includes any confirmatory testing. So if a variant of interest is identified in the analysis, if what additional methodologies need to confirm the presence of the variant, that will be done at CHEO. Our Reports will be issued, as are all of our current molecular genetic reports, through our lab information system, EPIC. So for providers who have EPIC access, you'll get your reports through EPIC. For those that don't, you'll get them through fax, like with all of our current reports. In terms of what is currently reported, the report is broken down into primary findings and secondary findings. For the secondary findings, we have different categories of reports, of variants, and that will be clearly indicated on the report. So related to the phenotype, that would be where we have a finding that provides a complete explanation for the patient's phenotype and is considered a molecular diagnosis for their condition. So those variants will be listed at the top of the report. Below that, we will list any variants identified that are possibly related to the phenotype. So suspected to be causative, but not sufficient to provide a complete molecular diagnosis. An example of that might be if we find one pathogenic variant in a gene that we know causes an autosomal recessive condition that is consistent with the patient's phenotype, but we don't have a second variant, or a finding that is a pathogenic variant that might explain part, but not all of the patient's phenotype. And then we will be reporting some findings in candidate genes where there is evidence of association with disease in humans, but has not yet made it to the known gene classification. So we are not searching for novel gene disease associations. This does not replace the work of research that is currently ongoing, but if there is significant or substantial evidence that a gene disease association exists, but it hasn't yet made it, say to OMIM, for example, if there's a strong case to be made, that's something that we would report. In terms of secondary findings, as previously discussed by Meredith, there's a variety of different options depending on the patient age and decision that they've made. But just wanted to clarify here that if somebody is having the actionable in childhood subset of secondary genes analyzed, that will look at 52 genes in total. If the full list is selected based on the matrix that we've come up with, then it will look at 59 genes in total, and that would be reported on the proband. For parents and family members, they do not get a separate analysis, so we're not looking at the full subset of CMG genes in a parent. We only check the relatives if a variant is found in the proband, similar to how other commercial labs are currently handling this situation. If a secondary finding is identified in the proband and the family member has requested to know their status for the same variant, then it will be reported both in the proband's report, and a separate report will be issued for the family member. So to make that a little bit more concrete, if the patient opts to have all secondary findings reported, we find a BRCA1 mutation, parents elected to have their status reported, and it turns out it's paternally inherited, the proband's report will indicate that the variant was paternally inherited, and then the father will also receive a report with his own name and his own genetic status on that report to allow him to enter the healthcare system and get any follow-up care he needs related to that secondary finding. And that will be issued through EPIC, our lab information system as well. In terms of targeted variant analysis, so once we issue a report, there may be additional testing recommended for the proband or family members, and that will be specified clearly on the report. But I wanted to spend some time on this to make sure that it makes sense, that if we identify a variant in the proband, there are different follow-up testing strategies for other family members, depending on what the variant is. So if we've identified a variant in the proband and we're not sure of its significance, it might be indicated to do additional segregation testing in other family members. For example, if we find an X-linked variant in a male and there is a maternal uncle available, segregation in that maternal uncle might be indicated. If the segregation is being done to help in the interpretation of the variant in the proband, that will be available at CHEO or at SickKids if it's a case that they've reported. So that is considered part of the GSO program. If there's a variant identified in the proband and we are clear on its pathogenicity, follow-up testing in other family members would not be considered to the benefit of the proband. In that case, it would be cascade testing in family members, either for predictive testing or for carrier status updates. That type of testing is not part of the GSO program, so that should be done at a laboratory that typically offers testing for that gene. If we're speaking specifically about secondary findings, most of the ACMG genes are available within the province. So it would just be a matter of testing for that at an appropriate laboratory as you typically would. So just to summarize here, there's a difference between segregation testing for the benefit of interpreting the variant reported in the proband and cascade testing for the benefit of testing other family members. And like I said, it will be clear from the recommendations section of the proband's report what scenario that specific testing falls into and whether that testing should be done at our laboratory or an appropriate laboratory that has that gene as part of their normal testing menu. I hope that's clear. I know it's a bit tricky to wrap your head around, so I can answer further questions about that later on if they are still coming up. Additionally, after the report is indicated, we will have the ability to perform reanalysis and reinterpretation. Typically, this will be available 18 months after the report is issued. And so if your patient remains unsolved, after 18 months, you could contact us to ask us for a reanalysis of the genome-wide sequencing data. In some scenarios, it might be appropriate to request an earlier reanalysis. And specifically, those scenarios would be if there's a new pregnancy in the family, if there's progression of the phenotype, or a new phenotype emerges in the proband, and the genetic testing status would have impact on the patient's medical management. And so upon request, those reasons for an earlier reanalysis could be considered. When we do a reanalysis, there's several different variables that could change between the time of the first interpretation and the reanalysis. There could be an updated bioinformatic pipeline that has changed in the interim. There may be updated annotations such as new genes described or new disease gene relationships known in the interim. An updated phenotype information could reprioritize or highlight different variants that weren't considered to be of interest at the first interpretation. And then reinterpretation, in general, there may be more information or different weighting of information available down the road. So those are some of the things that can change upon reanalysis. To request a reanalysis, there is going to be a separate requisition for that purpose. It will be available on our website. And basically, we're asking for standard demographic information, the referring healthcare provider, as well as an update on the phenotype or the family situation so that we have more information or confirmation of the previous information is still accurate. And so we will require that requisition in order to trigger a reinterpretation or a reanalysis, as well as an updated clinical assessment note so we know what's happening with the family at that point in time. And with that, I'm going to turn it back over to Kim to talk about some of the quality improvement opportunities, and then we will take questions.
Okay, thanks, Caitlin. Can you hear me okay?
Yeah.
Good. So it's just really brief, just finishing up here, guys. So we have lots of time for questions. There's essentially two studies that we'll talk about, and that's secondary findings and reanalysis. And in both those instances, the reason that we're examining this to this level of detail within GSO is because neither of them are currently funded. So the funding for secondary findings and the funding for reanalysis is currently not included as a funded test from the Ministry of Health. So in order for us to set ourselves up, just like we're doing with our examination of genomes versus exomes and their utility, to set ourselves up in the province for 2 years from now, and what is the best guidance that we can provide to the ministry on what the test should look like and what parts of it are reimbursed, we need to capture these outcomes with help from everybody else. So the first is secondary findings. And we actually have a really remarkable opportunity to do the best study internationally because of the way we can potentially operate within Ontario. So we all know that there's lots of secondary findings literature out there. It's often run and been done by folks at NIH and other groups. But what and they sort of give you a, you might find it in 1 to 3%. And we've all got experience over the last 5 years that we don't see them very often. What no one understands about secondary findings is what is the impact not only on the proband and the immediate family members, but on the extended family members as well, and what long-term or downstream consequences how does that impact the healthcare system, and what are the outcomes from that? So we went through and we looked at and projected the number of secondary findings that we think we will find within the 2-year project. And it's not very many, it's probably under 100. And what we're going to do is set up a study such that in collaboration with all of our different centers, we can actually capture the impact of the reporting of these findings, both with how much time it's going to take the lab to do it, how much time in the clinical end of the team does it take for us to deal with these and the downstream family members that may then be referred to see us, or we offer to see, and what is the healthcare system cost with all that, right? Because we have to make a case that this is an important piece of the test. And that's definitely what we heard from the clinical genetics community when we did the survey, which I think was about 6 months ago or so. So we're going to recruit both positive and negative secondary finding cases from all of our sites by invitation because we have that consent to recontact for research. We will set up and have Clinical Trials Ontario REB approval for all of our sites, just like we've done for many other studies we're all working on, like Care4Air Solve as well as EpiSign. So we're getting good at doing this with the multi-site approval, so that works. And then we'll collect this really, really valuable outcomes for this and be able to provide that back, not only to Ontario, but also to the international community at large, which will be really important. Okay, next slide, Caitlin. And then the other project that, of course, is going along with this is Care4Air Solve, which pretty much everyone is participating in. I did see some notes on the chat. People had questions around, I think, Lauren, you asked about looking at your own data for a negative case. And I think Andrea asked something about Care4Air Solve as well. So this still is running in parallel to GSO, and any patient studied in GSO can cross over to Care4Air Solve, which means then when they cross over, we can have access to their data through Genomics for RD, which allows us to have complete access and share that data in a more fulsome manner than compared to our knowledge base, which Meredith talked about a little bit. So all individuals can be consented into phase 1 optional, which is just sharing their deep phenotypic as well as all their genotype data, or phase 2, which is we've really looked at this particular patient. They are still unsolved. They can go into phase 2 and get other omics if they're eligible. And so we'll work with all of our individual sites that are participating in the CHIO arm of GSO, such that this workflow is well established. And when data moves to Care4Air Solve from GSO, all we will require is the Care4Air consent having been signed at the local site, no other data release forms. That's the advantage of actually having our data generated in the province. I think the other thing I just wanted to mention is the reanalysis. So we saw sort of the 18 months has sort of been this arbitrary, line in the sand that we've drawn and when reanalysis will be available. But we're also going to be capturing outcomes from reanalysis, because again, it's currently not a reimbursed test, but it's a really important aspect of the test. And so we do need to show why it's important and what the outcome of that is. So with respect to resources, you just heard from Caitlin and Meredith, and their contact information is there. And you can feel free to reach out to either of them to sort of clarify how things are going to go if there's some confusion. And then here's a list of all the resources. Again, we'll make this available so everybody can see the various places that one can reach out. And then just finally, just to wrap things up, just a huge set of acknowledgements to all these great folks on this slide from CHEO and SickKids that have worked really, really, really hard over the past, well, it's been almost a year at trying to get to where we are now, but even the years before that, as we were envisioning how this potentially might work and how to bring into Ontario something that is going to be remarkable and a value add and incrementally better than the service that we currently have, right? And you can imagine that is quite a set of aspirations. And so we certainly hope that we've set things up such that we have some unique things that we're going to be able to do because we're going to be performing this test in Ontario for the next 2 years. Okay, so now we have some time for questions and answers. So Caitlin and Mary, are you going to screen some of them?
Yeah, I'll take a look, I guess I'll start with the ones that are in the chat. I'll cherry pick the easy ones and farm out the more difficult ones to yourself and Meredith. In terms of, I saw a question, I think a couple questions about whether we can accept provider signature on behalf of the patient, given that many patients are seeing virtual right now. And yes, that is appropriate. As accepted by the MOH, a provider can sign on behalf of the patient's decision. In terms of sample requirements, this will all be available on the requisition as well as on the website. But for blood, we're requesting at least one EDTA tube of 4 milliliter size. Here we go. The question about if parents do not live in the province, is there a way to obtain the sample? So we could accept parental samples from out of province, but it would still have to be a blood sample. So it would be on the provider to coordinate with the parent or biological relative who's out of province to get a sample collected and sent to us. But the requirement is that the proband themselves has OHIP. If the parents don't have OHIP, we could still do the genome-wide sequencing as long as the collection is handled by the provider. Okay, I don't want to skip too many questions and then forget to go back to them. I'll throw this one out to Kim. A question from Dr. Sue. Will there be any options for doing WES for Amish and Mennonite families who do not have an OHIP number?
Good question, Vicki. In that scenario, are you expecting that they'll self-pay, or how do you think that would work?
No, they would not have the funding to pay self-pay. What we have been doing is sending down to the States to Clinic for Special Children. But it would be nice to be able to do some of it in Canada. But I'm just asking.
Yeah, I mean, I hadn't really thought about that, Vicki. Let's just take a little bit to think about that. When things get up and running and we sort of understand where we're at, I would, really like to see us devote resources to this type of challenge, as well as folks who are in Ontario with refugee status, right? Is there a way that we'll be able to help provide care for some of these folks, but not have but we may not have a mechanism for billing? Something I think to consider when we're a bit more established and have found some efficiencies in our work that will be able to absorb that kind of thing. So on the radar.
There's a question about supplying a positive control to another lab, for example, if familial cascade testing is being done. And yes, we can provide positive controls as we do right now. There's a question about turnaround time. I guess I'll take the first stab, is that our intention is to get results back within 12 weeks. That is what the current commercial diagnostic lab turnaround time is for routine priority cases. The benefit of having within province is that we don't have to go through the Ministry of Health approval process, which often adds significant time to that. However, as we're just getting up and running, there's a lot of variables that are either not in our control or we don't know about yet. So that's our intention. I don't know that that's chiseled on a stone anywhere. Is that a good answer, Meredith and Kim?
Mm-hmm.
Perfect.
Yep.
Okay. Question from Tooch. Is there a cap? Is there a maximum number of patients within these 2 years, perhaps funding-wise, that would halt the process? Kim, I think that's a question for you.
Yeah, that's a great question too. What's with the questions from London? Yeah, so we do have the idea of what the project volume should look like, guys, and they're quite generous. We did estimate, we watched how it grew every year, and we did estimate above that. We should all recognize that as soon as something's available in the province, the energy to get over the hill is a little bit less. But remember, we are still keeping it within the same practicing group of physicians. It hasn't opened up wide at this stage. Hopefully that will happen in the future. But Tooch, there is no cap per se. We are going to be reimbursed from the ministry on a per case basis. If it was to exponentially go quite crazy and be 3 times the volume we expected, that would be an issue. But I actually don't expect it will be, because the capacity of us as a group of clinics is sort of, what will be the rate limiting factor.
A couple questions about Care for Rare eligibility. First is, do patients have to go through GSO prior to enrolling in Care for Rare? And if patients have a negative GSO report, do they have to have over 18 months? I think that's meaning after the report, as opposed to the age of 18 months, to be eligible for Care for Rare. Meredith, do you want to take those questions?
Yeah, that's a good question. So no, you don't have to wait and do a clinical reanalysis through GSO prior to enrolling your patient in Care for Rare. So you can continue to follow the regular practice of, if you're seeing a patient, you have a negative result and you'd like to offer it to them at that time, then you can go ahead and enroll the patient, and we'll be following the same kind of research turnaround time.
Mm-hmm. And Andrea from Oshawa was asking about so Andrea, if the family, if the proband is eligible for ministry funded genome-wide sequencing, they would first go through GSO, and if negative, would come to Care for Rare. If there's something about that particular family that maybe would make them not eligible for ministry-funded exome sequencing, you could just reach out to us and ask that directly. But for most, I think we're at a place in Ontario where, all probands who should have genome-wide sequencing should have it funded by the Ministry of Health, and Care for Rare's role really is to look at those which are negative for novel gene discovery in the coding sequence, as well as other omics if that doesn't pan out.
Another sample question here from Dave, just to confirm, yes. So if it's a neonatal sample and 4 milliliters is not an appropriate sample requirement, we can accept 1 milliliter.
Dave is all about the details, isn't he?
Absolutely, he's keeping us on our toes.
Yeah.
Question from Gabrielle Lemire. What is the plan for large genetic panels that are currently sent outside of Ontario? Will they eventually be performed by GSO?
Yeah, these fellows of mine. Yeah, yes, Gabrielle. So I think that really is where I see this going. Right now, if your patient, my advice, if your patient meets eligibility criteria for genome-wide sequencing in Ontario, that is the test you should be doing if you're considering that versus a monstrously large panel. Those things just should move back into Ontario. But if for example, it's some sort of feature that is like, for example, say retinitis pigmentosa has quite a large number of genes. I think there's like close to 300. That's a single system. Currently, that's not funded, and that would still have to stay with a large panel. I do hope in the future, though, that the way we deliver diagnostic testing in this province will change, such that everything will go through sort of this common high-quality sequencing pipeline, and then sliced and diced at the local level in terms of for indications like retinitis pigmentosa. But we're definitely not there yet, and we have to do this 2-year pilot first.
Thanks, Kim. A couple more logistical questions. Question from Tuche about seeing that there's some patients that are within the Sick Kids catchment, but actually go to London for their genetic services, and wondering what the delineator there is. It's the provider or clinic address that determines where the sample goes, not the patient address. So if you're a provider in London, you always send your samples to CHEO, regardless of where your patient is from. Hopefully that'll make things a bit more streamlined. You don't have to pay attention to their address. Just know that if you're in London, you always send to CHEO. There was a question about urgent and rapid exomes. At this time, those are out of scope for the GSO program. So those will continue to go through the Ministry of Health Exceptional Access Program to be sent out of country. So
Yeah.
at this time, we're accepting routine priority samples only.
Yeah, and I think that we really do hope everyone that within a year will be able to transition that, but we have to, as you can imagine, we have to be operating at peak capacity and with well vetted pipelines, et cetera, to be able to absorb the rapid turnaround times that would come with those types of indications.
We got a question about the length of time for the committee review process. So I think that's referring to for cases where we're not immediately clear if they meet criteria or not, they'll be reviewed by a joint committee between CHEO and SickKids. We anticipate that you would be notified within about 2 weeks, just depending on what day of the week the sample arrives, what day of the week the committee meets. But we intend for that committee to meet weekly. So we should be fairly on top of that adjudication process. And if a sample a request is denied, or if we need additional information, you'll hear that relatively quickly after submission.
Yeah, it should be a faster process, everyone, than what it currently is with the Exceptional Access Committee, because that committee only meets once a month and we'd be meeting every 2 weeks.
Yeah. There's a question from Julie, just asking more details about the secondary findings study, wondering if we're intending to, or if it's possible, to look at penetrance of disease in individual with secondary findings as compared to penetrance of disease in the literature for patients or families that present symptomatically related to that condition.
Excellent question. So Julie, I think so, yes. Obviously, we're going to have some issues with numbers with respect to the number you'd have for the particular indication. But that's exactly why we want to do this in a systematic way for questions like secondary findings. It's also why we have set up this ReContact registry for research, so that questions like this, even deeper questions like this, with respect to all other types of indications, can be analyzed in more depth when we share data through Genomics 4RD and other ways, where we can actually then have a large enough data set that we can start to talk about things like penetrance and systematically ask it.
Thanks, Kim. There's a couple of lovely compliments in the chat. I won't read them out, but I encourage everyone to take a read if you're interested. There's one other question here from Jeanette that just came in. As many patients travel significant distance, would you bank DNA pending approval for the test? More convenient for patient and family not to have to travel twice. So that's a great question. The approach that we're taking right now is that we do not do prior approval process. So as this is a lab test, the whole process is initiated by us receiving a requisition and sample. We will extract DNA immediately and then use the DNA once the request has been approved. And so we don't have a prior approval process. If we need to put the testing on hold, or if it's rejected for some reason, we will keep that DNA. So if there's a long process of adjudication or an appeal, we'll still have access to that primary sample. But we do not want to get in the habit of receiving a sample with a request to do a DNA bank, and then later receiving a GSO requisition and accessioning a case for genome-wide sequencing. So we prefer just to receive the GSO rec and sample initially. We'll do the review process at that time and inform you if there's any issues in a timely manner. Question, will patients be notified if the test is declined? We'll send a lab report back to the provider that indicates that the testing has been declined for whatever reason, and then the provider will notify the patient as with any other test. I'm just noticing that we're at time now, so if anybody has additional questions, feel free to email them into myself or to Meredith at that gso@cheo.on.ca website. Or sorry, email address. We can also stay on the call for a few minutes if anybody wants to unmute themselves and ask a question. If you have questions that come up in the future, please feel to reach out to us. We're happy to engage and work through these scenarios. Our website will be up and running within the next couple of weeks, which will have the answers, I'm presuming, to most of your questions. So keep an eye out for that. Anything else to say to wrap up, Meredith or Kim?
No, thanks very much, guys. That was really helpful presentation. You did a lovely slide deck. which
Right,
I think
thank
will be
you.
available for everybody too, right? So
Yeah,
it'll
we'll
be-
put this presentation on the website as well. Okay, so thank you everyone to coming and for coming and for your engagement and interest in this topic.
Yeah, bye everybody.
Thank you so much.
Thanks, everyone.